Is that okay? I'm on faculty at Auburn University. So for about a decade, many investigators have been interested in the role of oxidative stress and life history variation. Yet oftentimes empirical studies really don't meet investigators' predictions. Today I'm going to tell you or describe some evidence that reactive oxygen species aren't always associated with damage, and I think that knowledge is particularly valuable for understanding the actual life history patterns that animals display, rather than always making the assumption that reproduction is costly. So the idea that reproduction has the idea that reproduction comes at a cost to future reproductive performance and longevity has been dogma in evolutionary biology for nearly a century. But why these processes occur has remained elusive. One hypothesized mechanism for this relationship is that females that reproduce will produce more reactive oxygen species than those that do not reproduce or reproduce to a lesser extent. So what this is during oxidative phosphorylation, you have electrons that escape the electron transport chain. They interact with oxygen and form what we call reactive oxygen species, or ROS, and those compounds can cause damage throughout the cell. So it's been thought that during reproduction, again, you could have elevated ROS production, and that could drive this relationship between current reproduction and future reproduction and reproduction longevity. But again, when we look across the studies that have been done to date, we find really mixed data on this. In fact, when we look at all of the data together, many investigators have now pulled data together and have shown that there's really no clear relationship between oxidative damage and reproduction. And in fact, not long ago, we conducted our own study on wild-derived house mice. We had animals that we kept until they were a year of age. We had animals that never reproduced, as well as animals that bred continuously until they were nearly a year of age. We looked at oxidative damage. We actually did find support for that idea. Those animals that reproduced did have higher oxidative damage. We also measured antioxidants. They showed that same pattern. So most people would stop there and go, higher damage, that should result in reduced longevity in these females. Oxidative damage bad, stop there. But we went one step further. We also measured the respiratory function of mitochondria. And what we found is that females that had reproduced had a trend toward improved respiratory function. You would expect with higher damage, you would have reduced function, and that was clearly not the case. So why would that be? Well, we now know, based on accumulating literature in the biomedical field, that reactive oxygen species are really important signaling molecules. Well, what do they signal? They signal the production of antioxidants, which quench damage or free radicals before they cause damage. They upregulate repair mechanisms that make damage go away. And they stimulate cells to produce more mitochondria. What does that do? That helps the respiratory performance of tissues. And so a new concept has been proposed called mitochondrial hormesis, or mitohormesis for short. And by this concept, when animals are exposed to modest levels of reactive oxygen species, this is associated with higher mitochondrial respiratory function, and that can lead to increased longevity. Whereas when animals have high levels of ROS exposure, that leads to that expected relationship that most of us think about when we see higher oxidative damage. And support for this concept comes from the calorie restriction and exercise physiology literature, and we think the same could also be true for reproduction. So if we go back to that data that we found in the lab, I think that when we found higher oxidative damage in animals that had reproduced, we could be in this beneficial range rather than in the damaging range. Okay? So if mitochondrial hormesis is responsible for this relationship, the data that I've described could have a broader role in the life history patterns of animals. And so we went back to the life history literature and we asked, as a community, are we really not approaching this problem in the right way? Usually when most people are interested in examining the cost of reproduction, they assume that the cost is immediate. It should have an immediate negative impact on the female. And if that's true, we should anticipate that a female's ability to allocate to reproduction should be reduced with each reproductive event. Of course, this particular pattern of reproduction is rarely seen. Okay? We still see quite a bit of variation. One common pattern we see is that similar to this sparrowhawk here. So let me zoom in on the sparrowhawk. So this is a relatively common reproductive pattern that we see in animals. It's definitely what we anticipate seeing in our mice. And what we typically find is that over a female's first few reproductive events, her performance increases. Then we see in the middle of the female's life, there's some variation, there's fluctuation, but there's relative stability. And then late in the life is when we see declines in performance, okay? So I think mitochondrial hormesis could be important in supporting this increase in high performance in the middle of an animal's life. Specifically, early in an animal's life, this process of mitohormesis can really be a process of energetic priming. In the middle of a female's life, a lot of animals, our mice in particular, are functioning near their physiological capacity. These mitochondrial hormesis processes could help keep her there. And then late in life is when we should really be looking for trade-offs, those negative effects. So today I'm going to focus on these potential positive effects of ROS on reproduction. I'll end with future directions, making some suggestions on how we should think about delayed effects in the future. So if mitohormesis contributes to life history patterns, we can make a number of predictions. All of these are things that we've looked at in my lab. I'm not going to go through them. Today, what I'm going to focus on is the prediction that a female, when animals reproduce, they should perform better when they're exposed to an oxidant in the future. Okay, so I like to think about this as looking at a sequential reproductive events, right? If one reproductive event is associated with modest ROS signaling that has beneficial effects, that ROS signaling should benefit the next reproductive event that also has modest ROS signaling, right? Of course, reproduction is a really complex process, and so what we did here was we bred animals, exposed them to an oxidant later, so we're really looking at how they respond to future ROS instead of all the other complicated things associated with reproduction. So we used our favorite model organism for this study, wild-derived mice. Lots of reasons for using mice. In particular, we use wild-derived mice because if you look at outbred strains of mice, they've been subject to artificial selection for high litter size, and that could have some effects on oxidative stress and reproduction. So here's our experimental design. We started the study with 20 animals that we reproduced, 20 animals that did not reproduce. All of the non-reproductive animals would be collected on an age-match basis to the reproductive animals. We bred the animals twice. The reason for that is in our mice, first-time moms, there's a lot of variance in their performance. We want to ignore that variance, and they usually pretty consistently do a good job second time. So after the end of the second reproductive event, 1 week later, we exposed animals to an oxidant. And what we did for that is we drove the mice over to vet school and x-rayed them. 500 rads of radiation for 150 seconds. We know that causes an acute increase in ROS. 4 days later, we euthanized the animals. We've done time course of experiments to look at how oxidative damage affects the organs, and we know 4 days later, the organs are starting to recover. So we're really looking at differential recovery. Okay, and then within each of those groups, only half of the animals were x-rayed, so we have that for comparison. So that gives us 4 groups. We have a reproductive control, a reproductive x-ray, a non-reproductive control, and a non-reproductive X-ray. We collected several different organs, and today I'm going to focus on our data for the liver and skeletal muscle. And of course, these are the most metabolically active organs in the body. Okay, so interpreting this data can be a little bit confusing, so let me talk you through it. In some cases, ROS is going to have negative effects. When those effects are negative, if we compare controls to X-ray, we should expect that animals that had reproduced should have a lesser consequence than those animals had not. And if the effects of ROS are positive, then we should expect that the beneficial effects, the difference between control and X-ray, will be greater. Okay. So we measure the respiratory function of the mitochondria by isolating them. We measure something called the respiratory control ratio. This is the ratio of the maximum function of the mitochondria by basal. And so that's RCR. And we can stimulate this either via complex 1 mediated oxidative phosphorylation or complex 2. Okay, so getting into our results. So first looking at RCR for the liver. What we found is that females that had reproduced had a significantly greater RCR after exposure to that oxidant. This was true for both complex 1 as well as complex 2. In fact, females that had not reproduced saw no benefit of that exposure. The same was also true for the skeletal muscle, particularly via complex 1 oxidative phosphorylation, and that was not true of complex 2. We looked at oxidative damage, and we found no difference in oxidative damage, whether we were looking at damage to proteins or damage to lipids, between any of our groups. So what are the take-home messages here? Okay, so we found that when animals reproduced, not only were they better able to deal with the oxidant, they had improved respiratory function when they were exposed to an oxidant in the future. So improved respiratory capacity. We found no difference in oxidative damage between our groups, and I think this really highlights the ability for cells to recover from oxidative damage. Reproduction in some cases is associated with elevated oxidative damage. X-ray we know is over the very short term. In the short period of time after X-ray and reproduction, when we collected our samples, that had gone away. So we think this process of mitochondrial hormesis may be associated with some of the beneficial effects that we see of reproduction on the performance of individuals. So that can be really valuable for thinking about differences in performance across the lifespan early in an animal's life. And sorry, I'm going over a tiny bit here, but I guess I started a little late. So what should we be thinking about late in life? Most of us, when we look at the cost of reproduction, we tend to be looking at animals that are relatively young or middle-aged, and we tend not to be collecting those old animals where we really should be seeing those effects. I think it's critical in the future to really be focusing in on these animals. And one thing that I think we really need to start thinking about is looking at mitochondrial DNA damage and mutations. So we know at the same time that these benefits are occurring, the mitochondrial DNA are slowly accumulating mutations. Cells have hundreds of mitochondria, and when you have damage to the DNA, mitochondrial DNA specifically, they're very effective at compensating for one another, and you need quite a high level of damage before it starts to have an effect on respiratory function. So I hope in a few years' time we'll be able to bring to you some of that data from the older animals. Thank you very much.